Welcome back to the deep dive. You know, if you ever really thought about the sheer resilience of biology, our bodies are constantly dealing with threats. Today, we're zooming right in, down to the smallest battlefield there is, the inside of a single cell, right when it hits a crisis.
Right.
And we're not talking about, the cell just dying immediately, not a guaranteed death sentence. We're looking at its really sophisticated last stand.
Yeah, that crucial moment.
Exactly, that moment where it's injured, it's stressed, kind of teetering on the edge.
Yeah.
But, and this is the key part, it still has this ability to potentially repair itself completely, to snap back.
That really is the core of what we're digging into today. Our mission, really, is to define and, dissect reversible cell injury. We call it RCI for short.
RCI, okay.
Yeah. And it's this kind of elegant, temporary state the cell enters when it hits a stressor. Could be lack of oxygen, maybe an overwhelming toxin, even physical damage.
Right.
The cell starts showing signs of distress, both functionally and structurally. You can see things are going wrong. But critically, it hasn't crossed that line yet. It hasn't passed the point of no return.
Okay, I like that analogy.
Yeah.
Thinking of RCI, like the cell is in the emergency room. It's, badly damaged. Systems are starting to fail. But the machinery, the body's systems, still have a shot at stabilizing it. And the defining thing about RCI, like you said, is if we can remove that damaging thing, the toxin, the lack of oxygen, right then.
If you can remove it in time.
Yeah, in time. Then the cell can actually undo the damage, reverse those structural biochemical changes, and get back to normal.
And that brings us right to this concept that's, well, frankly, high stakes in medicine, this idea of the point of no return.
That threshold.
Exactly. RCI exists right up until that threshold. It's a bit of a fuzzy line sometimes, but it's there. Cross it, and, well, the cell's fate is sealed. It's headed for death.
Either through necrosis, that messy breakdown.
Right, the inflammatory kind of death, or apoptosis, which is more like programmed self-destruction, more organized.
Got it.
And for you, the listener, especially if you're in medicine or biology, understanding this threshold is absolutely key. It helps make sense of how we treat everything from heart attacks, that's ischemia, to chemical poisoning.
We're basically studying the biological limits of repair, right? How far can the cell go before it just can't come back?
Precisely. What are those limits?
Okay, so we need to look for the early warning signs then. When a cell slips into RCI, the sources we looked at are pretty clear. There are two big features that consistently pop up first. They're like the flashing red lights telling us the cell's in trouble and fighting back.
Yes, these are the signature changes. You see them biochemically, you see them morphologically, meaning, under the microscope. The first one is the most universal. It's generalized swelling.
Okay.
The whole cell swells up, and its internal parts, the organelles, they swell too. It's a physical sign that the cell's energy system is basically collapsing.
And the second one?
The second is fatty change. This is an abnormal buildup of lipid droplets, fat, inside the cytoplasm. Now, swelling is pretty much universal in RCI.
Right.
But fatty change, that tends to be more specific. You see it mainly in organs that deal with a lot of fat metabolism. Think liver, primarily. Sometimes heart or kidney, too.
But that swelling,
yeah,
you said it's fundamental. We really need to understand what's causing that, because it seems like the first, most basic response.
Exactly. It's the core mechanical response to that biochemical failure we mentioned.
Okay, let's unpack that engine failure then. If the swelling is the visible crisis, what's the deep down biochemical reason? What's making all that water rush in? The sources are really clear on this. RCI almost always starts with events that cause a severe and often rapid drop in ATP.
ATP depletion.
So why is ATP, the cell's energy money, why is it so critical for just holding the cell's shape together?
Well, ATP is everything, functionally speaking. It powers all the systems keeping the cell stable and, well, alive. And the system that needs the most constant energy supply, the one that fails first when ATP levels plummet, is the Na plus MASK plus plasma membrane pump, the sodium-potassium pump.
Okay, I remember that one from basic bio, the pump in the cell membrane.
That's the one. Think of it like the bilge pump on a ship. It's constantly working, pumping water out to keep the ship afloat.
Right, against the tide.
Exactly. This pump uses a huge chunk of the cell's resting energy, maybe a third of its ATP, just constantly pushing 3 sodium ions out for every 2 potassium ions it pulls in.
Okay.
And this constant pumping maintains a really delicate balance of ions and osmotic gradient across the membrane.
So if the power supply, the ATP, suddenly tanks because of an injury, that bilge pump just stops.
Pretty much immediately, yeah. Or it slows down dramatically.
Yeah.
And the consequence is catastrophic. It's flooding. Yeah. When that pump fails, sodium ions, which are normally kept at a low concentration inside the cell, start rushing in, just following their natural concentration gradient.
Downhill, basically.
Right. And water. Water is an osmotic follower. Wherever sodium goes in high concentration, water rushes in right behind it, trying to balance things out. Osmosis.
Ah, okay.
And this massive influx of water is what causes that hallmark early sign of RCI. Rapid, generalized cellular swelling. The whole cell just blows up like a balloon, the membrane stretches, everything inside expands.
Wow. So it's literally an internal flood caused by an energy blackout.
Yeah.
That makes a lot of sense.
Oh.
Okay, so where does this energy crisis actually come from? What causes the ATP to fail in the first place?
Good question. The sources really highlight two main ways this pump failure gets triggered. The first one is probably the most common in clinical settings, things like stroke or heart attacks or even issues with preserving organs for transplant. It's oxygen deficiency.
Hypoxia or ischemia?
Exactly. Hypoxia is low oxygen. Ischemia is loss of blood flow, which causes hypoxia. Oxygen is essential. It's the final molecule that accepts electrons in the big ATP-producing pathway in the mitochondria. Oxidative phosphorylation.
And oxidative phosphorylation, that's the cell's main power plant, right? The high-efficiency one.
Precisely. It churns out a lot of ATP, maybe 32 ATP molecules for every 1 molecule of glucose. Super efficient.
Okay.
So when oxygen becomes scarce, that main power plant shuts down. The cell then tries to switch to its backup generator,
Which is?
anaerobic glycolysis, making ATP without oxygen. But the problem is this backup system is incredibly inefficient.
How inefficient?
It only makes 2 ATP molecules per glucose molecule compared to 32, not nearly enough to keep things running normally.
Okay, so a huge drop in energy production.
Huge drop. And there's another major problem with glycolysis running full tilt. It produces lactic acid as a byproduct.
Ah, right. So the energy crisis quickly turns into an acid crisis too, a double whammy.
Exactly. The lactic acid builds up, lowers the pH inside the cell, and that acid damages critical enzymes and just makes everything worse. It adds to the functional chaos and pushes the cell deeper into RCI.
So the ATP drop is from shutting down the main plant, and the backup system, while trying to help, actually makes the environment toxic.
That's a great way to put it.
Okay, and what was the second major way the pumps can fail?
The second pathway involves direct damage to the mitochondria themselves, so it kind of bypasses the need for hypoxia initially.
How would that happen?
Well, things like high doses of radiation, exposure to certain heavy metals, or specific biological toxins can directly injure the mitochondria.
So even if there's plenty of oxygen.
Right, even with oxygen around, if the mitochondria are damaged, they just can't run oxidative phosphorylation efficiently. They can't make enough ATP. So in both scenarios, lack of oxygen or direct mitochondrial damage, the end result is the same. The power grid fails, the sodium-potassium pump sputters out, and the cell swells up.
Got it. ATP depletion is the linchpin.
Yeah.
#tag B morphology on a macro and micro scale.
Okay, so we've got the cause, that ATP depletion, and the immediate physical result, the water rushing in, the swelling. Now let's shift to what RCI actually looks like. You know, what a pathologist or a clinician might see, both with the naked eye and under a standard microscope.
Right, the visible evidence. What happens if, say, a whole organ is affected?
Good question. If the injury is widespread, affecting a lot of cells in a solid organ like the liver or kidney, you can often see changes macroscopically just by looking at it.
Gross changes, they call them.
Yeah, gross anatomical changes. The organ will often look paler than usual. It displays power.
Why paler?
It's largely because all those swollen cells are actually compressing the tiny blood vessels, the capillaries within the tissue, so less blood flow locally makes it look pale.
Huh. So the cells get so swollen they effectively choke off their own blood supply, making things even worse.
That's a really good way to think about it, yeah. You also often see increased turgor.
Turgor, meaning?
Meaning the organ feels firmer, stiffer, maybe even a bit rubbery or swollen to the touch. It's lost its normal softness because it's waterlogged.
Okay. Stiff and pale.
And predictably, because of all that extra water, the affected organ will weigh more. It'll have an increased weight. So if you see an organ that's pale, stiff, and heavy, that's a big red flag for widespread, severe RCI.
Okay, now let's take a piece of that tissue and look closer. Under the light microscope, what are the classic signs inside the individual cells?
Under the scope, the most obvious thing you'll see is that the cytoplasm, the main body of the cell, looks kind of bubbly or cleared out in spots.
Yeah.
It's dotted with small, clear pockets or vacuoles.
Okay.
This is the direct visual evidence of that internal flooding we talked about. These aren't just random bubbles, though. They actually represent parts of the cell's internal membrane network, specifically the endoplasmic reticulum, or ER.
The ER. That's involved in protein synthesis, right?
Right, and also lipid metabolism. But here, sections of the ER have literally ballooned up with fluid because of the osmotic overload. Then they sort of pinch off, forming these small, clear vacuoles within the cytoplasm.
So the cell's internal plumbing system is getting stretched and fragmented by all the water?
Exactly. And because this pattern is all about water accumulation, hydro means water pathologists often use specific terms for it. You'll hear hydropic change or vacuolar degeneration.
Hydropic change. Vacuolar degeneration.
Seeing that under the microscope is undeniable proof that the osmotic pumps have failed and the cell's water management is completely overwhelmed.
Now, earlier you mentioned a color shift. When tissues are stained, typically with H&E, hematoxylin, and eosin,
Right, the standard pink and purple stain.
you said the cytoplasm starts to look redder or more eosinophilic. That seems kind of counterintuitive. Why would an injured cell turn more pink or red?
Yeah, it's a really important sign, so you have to remember what the stains stick to. Hematoxylin, the blue-purple stain, binds strongly to acidic things, especially nucleic acids like DNA in the nucleus and RNA in the cytoplasm.
Okay, blue binds to DNA and RNA.
Right. And eosin, the pink red stain, binds to basic things, primarily proteins in the cytoplasm and extracellular matrix.
Pink binds to proteins. Got it.
Now, a healthy, active cell has tons of ribosomes, which are made of ribosomal RNA, rRNA, scattered throughout its cytoplasm and attached to the ER. They're busy making proteins. All that RNA makes the healthy cytoplasm quite basophilic. It picks up the blue hematoxylin, giving the cell a kind of purplish tint overall.
Makes sense. So what happens in RCI?
In RCI, especially as the injury gets worse, several things happen. One key event, which we'll talk about more with the ultrastructure, is that ribosomes detach from the swollen ER. You get a loss of this ribosomal RNA.
Ah, so less RNA means less blue staining.
Exactly. Less blue binding. At the same time, some of the proteins within the cell might be starting to denature or clump together because of the stress and altered pH. These proteins bind the pink eosin dye more intensely.
So less blue, more pink. The overall effect is that the cytoplasm looks redder, more eosinophilic.
Precisely. And what's fascinating, as you pointed out, is that this isn't just a static sign of RCI. The sources note it's often progressive, meaning the eosinophilia becomes more pronounced, the cytoplasm gets pinker and pinker as the injury worsens and heads towards becoming irreversible, towards necrosis.
So simple swelling might be reversible, but that increasing pinkness is a bad sign.
It's a very significant sign. It visually signals a profound functional failure. The cell is losing its RNA, its protein-making machinery is falling apart. It indicates a loss of synthetic capacity that makes recovery much, much harder. It's like a visual gauge of how close the cell is to that point of no return.
Okay, that makes sense. Let's switch gears a bit from the water crisis, the hydropic change, to the other major feature you mentioned, fatty change, sometimes called steatosis, right?
Right, steatosis, particularly in the liver.
So while hydropic change was about water flooding in, fatty change is about fat building up, a chemical accumulation.
Exactly. Fatty change is specifically a structural buildup of lipids, mostly triglycerides, inside the cell cytoplasm. Unlike swelling, which can happen in almost any cell, this happens predominantly in organs that are major players in fat and cholesterol metabolism.
Like the liver. You mentioned that's the main one.
The liver is the classic example, absolutely. But you can also see it in the heart muscle, kidneys, and a few other places under certain conditions. It signals more of a metabolic injury rather than just the purely osmotic stress that causes the initial swelling.
Okay, so why is the liver so prone to this? And what exactly goes wrong in the metabolic process during injury that causes fat to pile up?
Well, the liver is the body's central processing plant for fats. Fats come into the liver mostly as free fatty acids. Once inside the liver cell, the hepatocyte, they can be handled in a few ways. Burned for energy, turned into cholesterol or phospholipids, or repackaged for export.
Export, how does that work?
To be exported out into the bloodstream for other tissues to use, the triglycerides have to be combined with special carrier proteins called apoproteins. This combination forms a package called a lipoprotein, things like VLDL, very low-density lipoprotein.
Okay, so fat comes in, gets packaged with apoproteins into lipoproteins, and shipped out.
That's the normal flow. Now imagine the liver cell gets injured by a toxin. Common examples are chronic alcohol abuse or exposure to certain industrial chemicals like carbon tetrachloride, CCl4. Even malnutrition or diabetes can disrupt this.
So where does the system break down when these toxins hit?
The breakdown can happen at several steps, but a few key points are often disrupted. First, the injury might cause more fat to pour into the liver from peripheral stores, just overwhelming the system.
Too much coming in.
Right. But, perhaps more critically, the toxic injury often directly impairs the liver cell's ability to make those essential apoproteins, or to attach them properly to the triglycerides.
Ah, so the fat arrives, maybe even more fat than usual, but the packaging materials, the apoproteins, are missing or damaged.
Exactly. The assembly line for lipoprotein export is broken. Without the apoproteins, the liver cell can't package up the triglycerides into VLDLs to send them out.
So the triglycerides just accumulate.
They just pile up inside the cell. You start seeing these distinct, often large, clear lipid vacuoles appearing in the cytoplasm. That's fatty change, or steatosis. It's a really clear visual sign that the cell's metabolic machinery for handling fat has seized up due to the injury.
And this, importantly, is still considered reversible, right?
Yes, crucially, it is usually reversible. If you remove the toxic insult, if the person stops drinking heavily, or the exposure to the chemical stops, the liver cell can theoretically repair itself, start making apoproteins again, package up all that accumulated fat, and ship it out. The liver can return to normal.
Provided the damage hasn't gone too far, hasn't hit that point of no return where membranes rupture.
Precisely. But the presence of those fatty vacuoles is a very stark, visible sign of reversible metabolic distress, distinct from the purely osmotic swelling. #D, ultrastructural alterations, electron microscopy insights.
Okay, so we've seen the gross changes, the light microscope changes like hydropic and fatty change.
Right.
But to really grasp the sheer intensity of the cell's struggle, the fine details of the damage happening inside during RCI, we need to go deeper, right? Beyond the light microscope.
Absolutely. For that, we need the power of the electron microscope, EM. It lets us see the subcellular architecture, the organelles, in much greater detail. And the sources point out that RCI leaves some really definitive signatures at this ultrastructural level. There are about 5 key signs.
5 deep clues. Okay, let's break them down. Where do we start? Maybe the outer boundary, the plasma membrane. We know it swells. But what does the EM show specifically?
Right. The EM confirms the swelling, but shows more specific alterations due to that strain and osmotic pressure. You see these characteristic kind of irregular balloon-like outpouchings on the cell surface. Those are called blebbing.
Blebs, like little blisters on the cell surface.
Exactly like blisters. You also see a general blunting or smoothing out of the cell's normal contours. But maybe the most functionally significant change visible on the EM is the loss of microvilli.
Microvilli, those are the tiny finger-like projections, right? Especially on cells involved in absorption.
Precisely. You find them on epithelial cells lining places like the kidney tubules or the small intestine. Their whole purpose is to dramatically increase the surface area for absorption or secretion.
So what happens to them in RCI?
The EM shows they often become distorted, shortened, retracted, or sometimes completely lost early in the injury process. The source material even includes a figure showing this in kidney tubule cells.
And why is losing microvilli such a critical sign of functional failure?
Because it means the cell immediately loses a huge part of its functional surface area. If it's an intestinal cell, its ability to absorb nutrients plummets. If it's a kidney tubule cell, its ability to reabsorb vital solutes or filter waste is severely compromised.
Wow, so the cell is functionally crippled almost instantly when RCI sets in, just by losing that surface area. Okay, what's next? The powerhouse. The mitochondria. We already know they're central to the ATP problem.
Right, and at the EM level, you see evidence of that distress. First, they generally show swelling, just like the rest of the cell, consistent with the water influx and maybe altered ion handling by the mitochondria themselves.
Okay, they swell up too. Anything else?
Yes, and this is important. You start to see the appearance of small amorphous densities within the mitochondrial matrix, the inner part.
Small amorphous densities. What are those? And why are they a dangerous sign?
They look like little fuzzy clumps. They're thought to be precipitates, probably made of denatured proteins, maybe some lipids, just early signs of disorganized cellular junk accumulating inside the stressed mitochondrion. Now, in RCI, they are small and relatively sparse.
And the size matters.
Hugely.
Yeah.
Because if the injury progresses and becomes irreversible, these densities become much larger, denser, often loaded with calcium. They're then called large flocculent densities. Those large densities are a definitive sign that the mitochondrion is toast, permanently damaged and the cell cannot recover. So seeing only small densities in RCI tells us the mitochondrion is struggling but hasn't completely collapsed yet. It's still technically reversible.
Okay, critical distinction. Now, the third major site, the endoplasmic reticulum, ER. We already know it balloons up, causing that hydropic change. What else does the EM show?
Yes, the EM confirms that marked dilation of the ER cisternae, the internal spaces. You can see how they swell and fragment, creating those vacuoles visible under the light microscope. But the really crucial functional consequence seen clearly with EM is the detachment of polysomes.
Polysomes. Those are the clusters of ribosomes, right? The protein factories.
Exactly. Polysomes are strings of ribosomes actively translating messenger RNA into proteins. They're often attached to the surface of the ER, making it the rough ER.
And they detach in RCI.
Yes. The swelling and stress cause these polysomes to literally pop off the ER membrane. And when they detach, the cell's main assembly line for manufacturing new proteins, including enzymes needed for repair or those apoproteins needed for fat export, just grinds to a halt.
Wow, that sounds really bad. If the cell can't make new proteins, it seems almost guaranteed to progress to irreversible injury if the stress isn't removed quickly because it can't even synthesize its own repair kit.
That's precisely why ribosome detachment is such a critical ultrastructural marker. The manufacturing facilities are shutting down. If the cell can't build or repair, its chances of recovery plummet.
Okay. Fourth observation. Accumulating wreckage. Something about myelin.
Yes, the myelin figures. These are probably one of the most visually striking findings on EM and RCI. They look like these weird, whorled, concentric, layered structures, almost like a fingerprint or a sliced onion floating in the cytoplasm.
World structures. What are they made of?
They're essentially aggregates of damaged cellular membranes, phospholipid precipitates. As the plasma membrane, mitochondrial membranes, ER membranes get damaged and break down from the stress, their lipid components can rearrange themselves into these stable, layered whorls in the watery environment of the cytosol. They're like little balls of membrane debris.
So even in the reversible stage, there's already significant membrane damage and wreckage piling up inside the cell that it hasn't cleared yet.
Exactly. It's a visual sign that while the cell hasn't completely ruptured its outer boundary, its internal membranes are already taking a serious hit. It really underscores the high stakes of RCI damage is accumulating.
Okay. And finally, the command center, the nucleus. Are we seeing the really bad signs yet, like the nucleus dissolving, karyolysis, or breaking into fragments, karyoxis?
Thankfully, no. Those dramatic changes, karyolysis, pinosis, shrinking, karyarexis, those are pretty unambiguous signs of irreversible injury, characteristic of necrosis or late apoptosis.
So what do we see in the nucleus during RCI?
The changes in RCI are more subtle. The EM might show nuclear alterations like some disaggregation or clumping of the chromatin, the granular and fibrillar components inside the nucleus.
Meaning the nuclear material looks a bit disorganized.
Yeah, it starts to look a bit messy, maybe slightly clumped, reflecting the overall cellular stress, the low pH, maybe enzyme dysfunction spilling over from the cytoplasm. But crucially, the overall structure of the nucleus, the nuclear envelope, remains intact. The genetic blueprint is still preserved, holding out hope for potential recovery if conditions improve. Okay, so we've mapped out all these detailed signs of RCI, from the whole organ down to the molecules inside organelles. Now, it's really important to place this state, RCI, in the bigger picture, on the timeline of cell injury and death.
Right, because it's not just an on-off switch, right? It's a process, a cascade.
Exactly, and RCI is that critical, potentially final stopping point before the irreversible plunge. It's the last chance for rescue.
So let's talk about that timeline. The sequence of events really highlights the urgency here.
It does. The sources lay it out clearly. Almost immediately after an injury starts, think of blood flow being cut off to the brain or heart cell function is lost very rapidly.
How rapidly?
Seconds to minutes in tissues like neurons or cardiac muscle with high energy demands, function just ceases. But here's the crucial part. Even though the cell isn't functioning, it might still be biologically viable at this point.
Functionally dead, but structurally still capable of recovery.
Precisely. And that entire window where function is gone, but viability potentially remains, that is the clinical window of reversible cell injury.
That's the window where interventions like restoring blood flow can actually save the cell.
Absolutely. That's why time is so critical in treating stroke or heart attack. Now, if the injury persists, if that blood flow isn't restored quickly enough, the cell inevitably hits that point of no return.
The threshold.
Right. And that threshold is fundamentally marked by the onset of irreversible biochemical alterations that truly cause cell death, things we can't undo. And importantly, this biochemical death, this commitment to die, actually happens before you see the really dramatic visual changes of necrosis under the light microscope or grossly.
So the cell is biochemically dead before it looks dead.
Often, yes. The visible changes of necrosis, the complete breakdown, the inflammation take time to develop, sometimes hours. But the decision, the irreversible biochemical failure, happens much earlier. That surgeon's clock or the paramedic's clock starts ticking the moment viability fails, not hours later when the tissue looks obviously necrotic.
Okay, so all those RCI changes we talked about, the swelling, the blebs, the detached ribosomes, the myelin figures, they're the visible evidence of this desperate race against time. The warning signs before that irreversible biochemical collapse.
Exactly. They are the morphologic footprint of that critical window, which makes RCI the prime target for any clinical intervention aiming to save tissue.
So we can use the features of irreversible injury, of necrosis, to really sharpen the contrast with RCI.
Yeah.
What defines that final irreversible state?
Right. RCI involves swelling, but potential repair. Necrosis is defined by absolute structural and functional collapse. The sources really emphasize two defining phenomena that tell you the cell has crossed the line.
Okay, what are the two key markers that confirm permanent damage?
First, the cell reaches a point where it has an absolute inability to reverse mitochondrial dysfunction. Even if you restore oxygen or remove the toxin, the mitochondria are just too damaged.
They can't restart.
They can't restart efficient ATP production. Those small amorphous densities we saw in RCI, they've likely progressed to those large, permanent, calcium-rich flocculent densities. The power grid is permanently offline. No amount of oxygen will fix it now.
Okay, irreversible mitochondrial failure. What's the second marker?
The second is profound disturbances in membrane function. This isn't just the blebbing or swelling of RCI. This is catastrophic membrane breakdown.
Breakdown meaning rupture.
Yes. Rupture and leakage. Critically, this involves not just the outer plasma membrane breaking down and leaking cell contents out.
Which would cause inflammation.
Exactly. But also the rupture of internal membranes, particularly the membranes surrounding the lysosomes.
Ah, the lysosomes.
Yeah.
Those are the cell's recycling centers, right?
Yeah.
Filled with digestive enzymes.
That's right. They're little bags of potent digestive enzymes. Acid hydrolases, proteases, lipases, kept safely contained within an acidic environment.
So what happens when they rupture during irreversible injury?
It's catastrophic. Those powerful enzymes leak out into the neutral pH environment of the cytoplasm and just start chewing everything up from the inside out. It's called autodigestion.
The cell digests itself.
Essentially, yes. This uncontrolled enzymatic digestion is a major contributor to the messy, disorganized appearance of necrotic tissue. And once those lysosomal enzymes are loose, there's absolutely no going back. It's like setting off a bomb inside the cell, a biological acid bath, you could say.
And you mentioned the outer membrane leaking too. That leakage of cellular contents, the enzymes, proteins, etc., into the surrounding tissue, that's what triggers the body's alarm system, the inflammation we see with necrosis.
Precisely. That leakage is the critical difference. In RCI, the membrane is stressed but largely intact. In necrosis, it breaks down, and all sorts of intracellular molecules spill out into the extracellular space.
And these spilled molecules have a name, right? They're important signals.
Yes, they're often collectively called DAMPs, damage-associated molecular patterns.
DAMPs. You should probably define those quickly.
Sure. DAMPs are basically molecules that should always be inside a cell. When they show up outside the cell, it's a major red flag for the immune system, signaling severe tissue damage or necrosis.
What kind of molecules are they?
Things like ATP itself, outside the cell, is abnormal. Uric acid crystals, certain nuclear proteins like HMGB1. Their presence outside the cell acts like a molecular distress flare, activating immune cells and kicking off a robust inflammatory response.
And this leakage. This directly connects to modern medicine, doesn't it? To diagnostics.
Absolutely. It provides a fundamental clinical application. Detecting these specific intracellular proteins that have leaked out into the bloodstream is the entire basis for many diagnostic blood tests used to find and measure tissue damage.
Like for a heart attack.
Exactly. The sources mention this explicitly. Finding cardiac-specific enzymes or proteins like troponin in a patient's blood is basically biochemical confirmation that heart muscle cells have died. They've gone past RCI, their membranes have ruptured, and their contents have spilled into the circulation.
Or liver enzymes like ALT and AST for liver damage.
Same principle. Elevated levels of those liver enzymes, transaminases, in the blood mean hepatocytes have ruptured. The RCI window closed, necrosis occurred. So these blood tests are essentially measuring the irreversible consequences of cell injury.
Okay, so let's try to bring this all together. To summarize RCI, you can think of it as the cell's really sophisticated but temporary alarm state. It gets triggered when some external stressor messes with its energy supply or its metabolic balance. It's a defense mechanism, but a chaotic one.
Yeah, highly regulated, but definitely chaotic. But the key is, if that damaging thing, the toxin, the lack of oxygen, is removed quickly enough...
Before the point of no return.
Right. The cell actually has the potential to hit the reset button. It can restore its ATT levels, fix the pumps, repair the swollen organelles, clear out the debris like those myelin figures, and get back to business.
The core takeaways here seem pretty fundamental for understanding, well, almost any disease process, really. We've learned RCI is mainly defined by maybe 3 big things.
I think that's fair. Takeaway number 1. RCI's hallmark is cellular swelling, that hydropic change, and it stems directly from ATP pump failure leading to water influx. That's the basic mechanical crisis.
Okay. Number 2. Under the microscope, we look for specific signs of this struggle. Those clear bubbles, the vacuolar degeneration.
The swollen ER bits.
And that increasing thickness, the cytoplasmic eosinophilia, which tells us the cell is losing its vital RNA and protein-making ability. A bad sign if it gets worse.
Right. And maybe most importantly, takeaway number 3. RCI represents that absolutely critical window of opportunity. It's the last chance saloon for clinical intervention.
The final stop before the cell crosses that point of no return.
Which, biochemically, we now know means irreversible mitochondrial collapse and catastrophic membrane rupture, especially of the lysosome.
You know, that transition from reversible to irreversible, it's so starkly different depending on the type of cell involved. The source material had a really great comparison that drives this home.
Oh yeah, the variable consequences.
Yeah.
It perfectly illustrates how RCI's outcome is dictated by what the cell does and how much energy it needs constantly.
Right, they contrasted skeletal muscle with heart muscle.
Exactly. Think about a skeletal muscle cell in your arm. If you cut off its blood supply ischemia, it enters RCI. But skeletal muscle can often be rested. If you're not actively contracting it, its energy demands drop quite a bit.
So it has more time.
It generally has a longer window of RCI. It might survive for hours without blood flow before the damage becomes irreversible because its baseline metabolic rate isn't outrageously high if it's not working hard.
The heart muscle cell.
Yeah.
Total opposite. It has zero luxury of resting.
None whatsoever. Cardiac muscle cells are contracting constantly, pumping blood, demanding huge amounts of ATP second by second without a break. Their metabolic demand is extraordinarily high and continuous.
Which means their ATP reserves are tiny, and that point of no return comes much, much faster.
Drastically faster, often measured in minutes, not hours. If a coronary artery gets blocked and a section of heart muscle loses its blood supply, significant irreversible damage, necrosis, can start within just 20 to 30 minutes.
Wow, 20 minutes.
Yeah, that's when you start seeing those cardiac troponins leak out. The heart simply doesn't have the metabolic cushion or the option to rest and wait for rescue like skeletal muscle might. Its vulnerability stems directly from its nonstop essential job.
That's a fantastic illustration of how context matters. So we'll leave you, our listeners, with a final thought to chew on, building on that idea of vulnerability based on function. If the outcome of RCI hinges so much on metabolic demand and how critical the cell's job is, what other cell types in the body do you think might be right up there with cardiac cells in terms of having a really low threshold for irreversible injury, maybe neurons in the brain, or those rapidly dividing cells lining the gut? And more importantly, why might understanding that specific vulnerability be so crucial for tackling human diseases affecting those tissues? Something to think about.